Hi class, welcome to your Module 8 lecture. The topic for Module 8 is membrane structure and function. So we're going to talk about the different components of the cellular membrane, but we'll also begin our discussion on energy. Before we get ready, I know some of you are meatball fans in my class. He's right behind me. There he is. Well, he's got his back to you, he's sleeping. So this is content from chapter 5 of your textbook, and it's the entire chapter. All right, so we'll begin with the discussion about membrane structure and function. So we'll spend some time reviewing the different types of proteins that are present within the cellular membrane. And then we're going to begin our conversation about energy. And this will be a conversation we continue in subsequent modules. We will wrap up today's lecture by talking about enzymes and how they work, what their functions are. So the membrane is described by the term the fluid mosaic model, and what that describes. The word fluid implies that it's got some movement and perhaps that it's made out of fluid, which both of those are accurate. But the mosaic part of this term refers to the presence of many different proteins that are present within the cell membrane. So it really is correctly described as a patchwork of different proteins that are present in those phospholipids. So we've already learned about our different types of biomolecules. We learned about lipids, and one of those types of lipids are the phospholipids. And these are the specific type of fat molecules that make up the cellular membrane. And in it are the different proteins. So it is described as being selectively permeable. And what that means is that the cell membrane does regulate what goes through it. And we'll learn about how that happens. We already know that proteins are specific molecules. They interact through shape. And since there's protein in the cell membrane, that's a great part of how it is. It's about the matched protein binding to the correct protein in the membrane. And the different proteins that are embedded within that phospholipid bilayer do have different functions. So we will spend the first part of this module reviewing the general functions of the different categories of proteins that are there. All right, so here's a figure from your textbook, and we're looking at a cartoon drawing of two cells that are interacting with each other. And we can first of all focus in on the phospholipid fat molecules that make up the actual membrane itself. And then we're going to turn our attention to the various proteins. So just to remind you, the membrane has two layers. That's why it's called a bilayer. And they are oriented opposite to each other. So the water soluble part is on the outside and on the inside. And then in between is where the fatty tails, that fat part is. So looking here, we've got a number of different proteins that are dispersed through the membrane. And each of these represents a different category of protein, and we will learn those. We'll learn their names, but more importantly, we'll learn about the functions of these different proteins that are present. So let's also just take a minute to peek inside. Inside the cell, we are seeing the microfilaments of the cytoskeleton. And some of these proteins are actually anchored to members of the cytoskeleton. So we'll review that as we go through. All right, so same figure here, but now the different proteins are labeled. so that you're able to appreciate the names of them. So I wanted to provide you both figures so that you have the blank figure here to work with studying. You could label it or quiz yourself on it. Now we'll review them. So first of all, we're going to begin with the process of diffusion, and we'll continue this discussion in a moment. We'll come back to it. But the diffusion is a process that does not involve, in this case, we're not seeing any involved protein. So the respiratory gases, which are molecular oxygen, O2, and carbon dioxide, CO2, they diffuse through the membrane. So diffusion is the mechanism for the movement of respiratory gases. That's for us, but it's true for other organisms as well. But also there are some other molecules that are able to move through the membrane by diffusion. The characteristic of molecules that go through the membrane by diffusion is that they're small. They've got to be pretty small molecules, and they're nonpolar. So let's look at the example of our structure, since we've got a nice enlarged picture to look at. The gray on either side of the membrane, those are the glycerol heads, which are hydrophilic, water soluble. And inside are the two layers of the phospholipid tails. So they have fatty acid tails on them, which are fat soluble. So the structure and function of the membrane is actually simple, but so powerful, really. And let's begin by having a little discussion about evolution. This structure of the membrane is the same as what is found in bacteria. It is what is found in archaea. It is found in the most primitive organisms. It is a structure that works. There has been selective pressure for it to become, for the development of different proteins that serve different functions. But for the most part, it hasn't changed much. It's the same basic structure as it's always been. And this tells us it's a very efficient structure that works. Now, if the membrane were all made out of glycerol head or without any fatty acid tail part, or if it were all hydrophilic, water soluble, it wouldn't even be there. It would dissolve in the watery environment that is either inside our cells or outside the cells. And if it were all fatty tail, if it were all hydrophilic, that would make it really challenging for different materials to move through it. Materials would just get stuck in the fatty acid part without moving through. I mean, we are water soluble. I mean, aqueous is a term for solutions that means that that the liquid is water, and we are aqueous. So we are in that watery environment. So this function is tied directly to the structure here, where we have the watery parts on either side of the membrane, where the watery parts of our body are, and then nestled in between, really protected, are the fatty acid tails. So the respiratory gases and a few other small nonpolar molecules are able to move through by diffusion, just squeezing directly through, following a gradient. We are going to come back to the concept of diffusion in just a moment. Now, molecules that are charged or polar are not going to be able to diffuse through. They're going to just get stuck. They're going to bond. Think back about the example of hydrogen bonding of one water molecule to another. Those bonds happen. So an ion's not going to be able to get through because it's going to just stick. Okay, we're now going to move on to the first category of proteins that are found in the eukaryotic cell membrane. And these are what are commonly called transport proteins. And like the name suggests, they have the function of transporting molecules across the membrane or through the membrane. We have two types that we're looking at, and both of them are shown in this figure. We're going to begin first with the channel protein that operates by diffusion. So the way I like to think about channel proteins is they're essentially a tunnel that connects the two watery environments, the inside of the cell to the extracellular fluid. And we see this blue, whatever it is, this blue solute moves through this channel protein. So these are proteins, and we know now that proteins are very specific molecules, or have the ability to be very specific molecules. So different channels are selective. They allow usually only the blue solute through in this example. The red one's not going through it. So this is a channel protein, and channel proteins use diffusion. They are one of the types of transport proteins. We'll now turn our attention to the right side of the figure, and we're going to look at now the active transport protein. This uses a different process. It is not diffusion. It is active transport, which for all intent purposes has a lot of opposites to diffusion. We're going to cover those in a moment. The big one is that active transport uses energy. So that means that it costs the cell in the form of energy to move through whatever it's passing through the active transport protein here. So ATP is the energy currency of the cell. That's energy. So we've got two different color solute molecules here. We've already talked about the blue one, and it is associated in this example with the channel protein, the diffusion protein. But this red one is going through the active transport protein. And since we're talking about proteins, they have the ability, again, to be very specific. You're not seeing the blue go through the active transport channel or vice versa. So transport proteins do allow specific molecules and even ions to enter or exit the cell. If it uses energy, it's active transport. If it uses the concentration gradient, that's diffusion. All right, we have a number of different enzymes that are embedded within the cell membrane. And we'll spend quite a bit of time today talking about enzymes and how they function. In this example, we're looking at a sequence of enzymes, a pair of enzymes, and there is an order of this reaction. So it begins with the initial reactant here, this yellow triangle, and it bonds, has the right binding site for this first part of the protein. Notice that the binding site is the opposite of a triangle to receive it. So the yellow triangle binds to the enzyme, and we have a chemical reaction happen where the orange circle is produced. So this is an intermediate product, but it binds to the second part of the enzyme. Notice the different shape in the binding site on each of these enzyme proteins. So the orange circle binds on to the second part of the protein, matching the shape, and the final product is produced, which is this green square. So this is a sequence of enzymes, and many of the reactions in our body actually are even more than just a two-member sequence. There are several enzyme cascades in our body that involve multiple different members of the relay. So some of the proteins that are in the cell membrane are enzymes. And we're looking here at the example of a sequential reaction. We will come back to the topic of enzymes in the last part of today's discussion. All right, next in our discussion of different types of proteins in the cellular membrane are what are called attachment proteins. So the key thing about attachment proteins is they are attaching to something. The name is telling us. So we're looking here at attachment to the cytoskeleton inside the cell. And then even up here, we're looking at attachment to whatever this yellow, purple protein is. So attachment proteins either attach to the cytoskeleton or to something in that extracellular matrix. And we're seeing examples of that, both of those things happening here. So these tend to be more anchored in place in the membrane because they're attached to something. The other proteins that we've looked at so far, they haven't been anchored to anything. So they move more freely in the membrane, where these are a bit more tethered. Because of that, there is support provided to the membrane by the attachment proteins, since they are anchored to the cytoskeleton or extracellular matrix. They provide physical structural support to the membrane. The attachment proteins, since they are spanning that membrane and are interacting with both the extracellular environment as well as the intracellular environment, they are often involved in sensing and coordinating responses to changes between the inside and the external environment. All right, so now we're looking at what is called a receptor protein. And receptor proteins, similar to what we just looked at, we have a signaling molecule that is the correct matched partner for this receptor protein. And that's based on that elaborate three-dimensional structure that we learned about for proteins. So the signaling molecule binds to the receptor. And in this case, it sends a signal to the inside of the cell, but the signaling molecule itself is not going inside the cell. So this, the water-soluble signals that our body uses, they tend to bind to receptors that are like this, where they stay outside the cell. The signaling molecule doesn't come in. So it binds to the receptor protein, and then a message is sent inside the cell, resulting in some function. It could be the production of another protein or the activation of an enzyme. So these are specific reactions and interactions, because now we're talking about not just one protein in the receptor protein, but many times that signaling molecule itself is also a protein. So protein to protein interactions are specific, and they are governed by that three-dimensional structure that is formed by the elaborate folding that proteins go through. So the receptor protein is bound, and then it signals inside like a relay. All right, these are examples of junction proteins, and junction proteins are a bit different from everything else we've looked at so far. Now we've got direct binding of cell to cell through the junction proteins. So notice that they match in shape. So we do still see specificity, and we would expect that since we're talking about proteins, but they are forming an actual direct contact between these two cells. I'm going to back up for a moment. The signaling molecule is released. So this is not a direct contact interaction, because the signaling molecule has been released from the cell that produced it. So the junction proteins are direct contact from cell to cell. So they do form those important cell junctions or intracellular junctions that are needed to make tissues. I mean, so far we've been talking about cell to cell, but a tissue is a collection of cells that are attached together by junctions. Now we're looking at what is called a glycoprotein. So this is still direct contact, because the molecule, they're bound to the cells that are interacting. So what a glycoprotein is it's a protein with a sugar group. The glyco refers to the presence of a sugar on the protein. So what is actually binding into this second protein is the sugar itself. I would like to emphasize that sometimes the glycoprotein and the protein receptor that binds it, it's not always just to the sugar group. Sometimes there's a binding to the protein as well. It just depends what specific case we're looking at. But these are direct connections from cell to cell. The way that glycoproteins work effectively in our body is they work as ID tags. So it's a way for the cell to know what cell it's communicating with and to verify that correct connection, if you will. So it's possible that the glycoprotein is recognized by other cells too, or on other cells. Anything that binds to the sugar part would be able to interact. So that gives the ability of the body to respond in different ways using the same molecule or part of the same molecule. All right, so putting it together, now we've got where we started. We've got diffusion of the respiratory gases and other small molecules, nonpolar molecules. They move right by through diffusion. We've looked at the example of proteins that are enzymes that are present in the cellular membrane, and we looked at a sequence. We looked at attachment proteins that really do bridge the external and the internal world. And that is because inside the cell, the attachment protein is anchored onto the cytoskeleton, where outside the cell, the attachment protein is interacting with something in the extracellular matrix. So this is cell to cell communication. This is how it's done. We're looking at the example of the receptor protein here, where we have this candy corn looking protein signaling molecule that binds to the matched receptor. And then once the receptor is bound by its correct partner, it sends a signal inside the cell to do something. We've got our channel. So this is a channel protein that allows for the diffusion of a specific molecule or group of molecules through it. And then right next door, we're looking at the example of an active transport protein. And this time it uses energy. We're going to talk about the differences between diffusion and active transport next. Our junction protein is a direct cell-to-cell contact between two adjacent cells. And this is one of the junctions that create tissues by bonding together cells that touch each other. And then we've got similarly, but this time we've got the glycoprotein with the presence of a glyco group or sugar group, which is interacting with its matched protein. So these two are a type of junction since it's direct contact, but the function of glycoproteins is usually as a cellular ID tag so that cells know what they're interacting with. All right, so that sums up the first part of the module. So diffusion is a process that does not utilize energy. It uses what is called a concentration gradient. And this is simply refers to the presence of where the molecules are. Let's take a look. So the gradient is always from higher concentrated to lower concentrated. So when we say that something is diffusing along the concentration gradient, it's moving from where there's more of it to less. So imagine if I had a bottle of perfume and I sprayed that. If you're near me, you would smell it probably right away. But if you're, say, in my kitchen, which is back there, it would take some time before the person in there could smell the perfume. because that's what diffusion does, is it begins where that highest concentration is, and then the particles move away until they're finally even. So we'll look at some better examples, but that's a good real life example to really to understand diffusion and gradients. I'll give you another real life example. This one is biology. And sorry, but I'm going to make you laugh. And this is because I had, a lot of boys in my life. Yes, a fart. Smelling a fart would also be an example of the application of diffusion. And hopefully you're far enough away from it where you won't smell it, right? All right. So diffusion is properly called passive transport, not active transport. And what happens is finally the particles will hit what is called dynamic equilibrium. Let's take a look at that. All right, so let's begin on the far right side of the figure here. So we have a membrane in the middle, first of all. That's what this gray dotted line represents. There are pores in it. So this is actually an artificial membrane. We would be doing this in the lab had it not been for COVID, where you utilize some artificial membrane and look at diffusion. But what happens here is that the holes, the pores, are big enough for this molecule of dye to move. So the gradient is always high to low, and that dictates which way the molecules are going to move. So the gradient is much higher on the left side of this membrane than it is on the right in the beginning. I mean, there's none on the right side at all. So we have a sharp gradient, a lot to none. I mean, you can't get much more sharper than one side having none. So over a bit of time, those molecules of this gold dye are going to move through until they hit their dynamic equilibrium. And at that point, the process is done. There's no gradient to push diffusion. Diffusion only works with a gradient. So therefore, when we hit equilibrium, there no longer is any gradient whatsoever. So the diffusion process stops at that point until the gradient reappears, if it does. So that is diffusion. It is passive transport because a cell does not need to use energy to do this. It just happens. Much like what happens if a person sprays perfume in the room, it's going to disperse through the process of diffusion. So what happens though if there's two different molecules? In reality, in our cells, gosh, there's lots of different molecules that each of our individual cells live in. So let's complicate it, or in other words, make it more realistic to what's actually happening in our body. So the gold or orange solute here is the same. It starts out in higher concentration on the left side of this membrane than it does on the right, and it diffuses along its own gradient, like we just saw. But let's add in the purple dye. So it is in high concentration on the right side of the same membrane associated with the gold or orange dye. So we've got two different gradients. What happens with one gradient has no bearing on the other gradient in this example. So let's see. So now the direction of diffusion for the purple molecule is opposite the direction for the gold one. And it eventually reaches its own dynamic equilibrium. And this may happen at the same time. It may happen at a slightly different time. So what I'm trying to impart upon you is that each molecule has its own gradient, and each molecule follows its own concentration gradient. All right, let's play this animation so you're able to see a bit more of this. I don't know that you have access to these animations. They are part of the electronic textbook So I do like to include them because I think that they have some good teaching points associated with them. All right, here we go.
Diffusion is the tendency for particles of any kind to spread out from where they are more concentrated to where they are less concentrated. This process can also be described as molecules moving down their concentration gradient. Diffusion across a biological membrane is called passive transport, since the cell expends no energy to move the molecules. Oxygen and carbon dioxide molecules move into and out of cells by passive transport.
All right. Okay, we're going to look at another animation here. And we saw a little bit of this in the video just now, but this demonstrates the selectivity of the membrane. And this ties to a lot of things, including whether or not there are protein channels that are being utilized for diffusion. So this is another short video. Maybe not. That was weird.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, they have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis.
Let me pause that there. So not only did that video segment review the process of diffusion, but it also looked at the example of facilitated diffusion, which is when it involves a channel protein to move through. So the diffusion of water is facilitated by the presence of a protein that is in the cell membrane that allows for the water to move through it. Water molecules can go through the membrane on their own, but it's slow that way. Instead, the majority of the water moves through a special protein that is made for that purpose. So what we've been talking about thus far, these have all been examples of diffusion. So regular diffusion or simple diffusion, facilitated diffusion, and osmosis, which is the example of the movement of water in response to a gradient. Now we're going to shift gears for the last half of this video segment and talk about active transport. This is not diffusion. It uses energy. Let's take a quick moment just to look at some of the features here. So this is a cartoon of the intestine, which is where the majority of the absorption of nutrients happen. Look at these weird peaks in it. These are wiggles in the membrane. And what this does is it increases how much membrane there is. And now the cell's able to utilize all the different materials that are on this extension of the membrane. It allows for the absorption of more nutrients into our body so that we can get the most out of the food that we eat.
The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
Okay, I'm actually going to pause that here so that we don't get too far ahead of ourselves, but I will return to the last bit of it in a moment. Okay, so diffusion again does not require energy, which is why it's a passive process. We have simple diffusion as well as facilitated diffusion, but both follow the gradient. The respiratory gases, oxygen and carbon dioxide, this is the only way that they move through our cell membranes. And that's really important so that oxygen may be delivered to every cell in the body and carbon dioxide may be removed. So why is it important that the movement of our respiratory gases be a passive process? This would be a great discussion that we would have in the classroom at this point, where you would discuss it with each other. But what happens if we don't have enough energy and we are using an energy dependent process? I mean, we've all felt that in terms of fatigue, you know, when perhaps you're training for a marathon or, you know, doing some regular form of aerobic exercise. We've all felt the consequences of when we need more nourishment in our body. So the reason why there has been evolution in the human lineage for the respiratory gases to move by diffusion is because there are times where we might not have enough energy, and we wouldn't want such a critical process dependent upon the process of active transport that just doesn't happen if the conditions aren't there. Also, as you progress through your different biology classes, really when you get into physiology, you'll begin to understand the whole body is set up, really. The cardiovascular system, the way that it's designed, the structure of it, is it's designed to where we have these gradients in place for the respiratory gases. So it works. It's not a problem. You don't hear of people having issues with, well, actually, I'm going to take that back because there are some medical conditions. I was going to say there aren't medical conditions associated with problems with diffusion of the respiratory gases, but there actually are if the surface where that diffusion happens is damaged. So osmosis is the specialized case of diffusion, and it refers to the movement of water following the gradient. So what's moving is the water, not the solute. So we'll look at this example here. There's actually a lot of great information packed into this one figure. So we're going to take some time to talk about it. And then I'm actually going to play you another video that reviews this figure because understanding this is the basis for many of the concepts that come next. So we're going to begin up top looking at this U-shaped pipe or tube. It does have a membrane at the bottom. And note that these solute particles, these purple solute, they can't fit through. In this case, the membrane is too small. It won't allow for the movement of the dye molecules or whatever it is. So we begin with an equal amount of water on either side of this membrane. So 1 unit of water to 1 unit of water. Let me get a box to make some notes on here for you. So we have, so if we just to give us some generic terms, so we have 1 unit on the left, and then we have 1 unit on the right. They look equal, right? They look to have the same volume. But let's look at the dye now. That is not the same. If we look at the number of dye molecules on the left, we've got what? 1, 2, 3, 4, 5, 6. So we have 6 units on the left. And then how many on the right? 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, it looks like 13 units on the right. So that's how we start. Oop, sorry, I went too fast, as I often do. However, what happens after osmosis has occurred is the water moves. So the particles, again, don't move here. So let's run through the same equation and look at the relative ratio so to hopefully appreciate this. So now on the left, say it looks like about half our water moved, right? So we've got 0.1 units of water on the left versus 1.5 units on the right, because half of it moved over roughly. And then our solute is still the same. So it's 6 units on the left versus 13 units on the right. But if we look at the ratio between these, it's evened out by the movement of the water. So sorry, let me just get back my mouse. It always disappears. So by moving over this amount of water, what it's done is effectively distribute it the same. So if we take 13 divided by 6, we have the same ratio as over here, but now our water's moved. So if we were to look at a value called molarity, which you've looked at in lab, that would allow us to see that it's equal because the water has adjusted by moving. So now let's zoom in. So now we're zooming into this black box area at the very bottom of the reaction. And this is before osmosis has occurred. So keep in mind, it's before osmosis has occurred. So what we're looking at are the molecular interactions that are present. So we've still got our dye molecules. We've got 1 on the left and 3 on the right, since we're just looking at this square. But look what's happening with the water molecules. They are hydrogen bonding to the purple solute. And if we take a step back and remember our chemistry from our earlier modules, we know that hydrogen bonding happens in watery environments to something that's charged, and it's always between opposite charges. So it looks like in this example, the hydrogen part is binding onto the water. So the water molecule does surround the solute, but you can see that the water molecule by itself is able to fit through. Let me get rid of all my drawings here. All right, move forward. All right, let's play this video.
Osmosis. Osmosis is the diffusion or net movement of water molecules across a selectively permeable membrane. An example of osmosis is illustrated here in an artificial system, a U-shaped glass tube with a selectively permeable membrane. The tube contains water, shown in blue, and solute molecules, which are sugar molecules in this example, shown as green dots. The drawing in the upper left shows the starting conditions for this example of osmosis. and the drawing in the upper right shows the conditions after osmosis has occurred. The drawing below shows a magnified view of the details of what's happening on both sides of the selectively permeable membrane during osmosis. Let's take a closer look at our artificial system. The U-shaped glass tube is divided in half by a selectively permeable artificial membrane. The membrane has pores that are large enough to let water molecules pass through, but too small to let the sugar molecules through. It's called a selectively permeable membrane because it allows some substances, like water in this case, to pass through, but blocks others like the sugar molecules. Notice that there is the same volume of solution on both sides of the tube. But there are fewer sugar molecules on the left side and more on the right side, meaning that there is a lower concentration of sugar on the left and a higher concentration of sugar on the right. This difference in sugar concentration across the membrane means that movement of substances will occur to equalize the concentrations. Let's look more closely at what's happening near the membrane. The membrane is shown as a gray dashed line. The water molecules are shown in red and white, and the sugar molecules are still green, but have more detailed molecular shapes in this enlarged view. Recall that the membrane will let water molecules pass through, but not sugar molecules. Notice that some of the water molecules cluster around the hydrophilic or water-loving sugar molecules. These water molecules don't move around freely in the solution. This means that the more sugar that's present, the fewer free water molecules there are to move around and diffuse. The right side has more sugar molecules and fewer free water molecules. The left side has fewer sugar molecules and more free water molecules. This means that there is a concentration gradient of free water molecules across the membrane in the opposite direction of the sugar concentration gradient. If the membrane barrier weren't there, the sugar and the free water would both diffuse, each down its own concentration gradient, until the concentrations on both sides were equal. But the membrane is there, and only water can pass through it, not sugar. What we have here are the conditions that cause osmosis. The free water molecules will move across the selectively permeable membrane, down their concentration gradient, until there's no longer a concentration difference of free water molecules or sugar molecules across the membrane. In this example, which way will the free water molecules move during osmosis? During osmosis in this example, the net movement of free water molecules will be to the right across the membrane down their concentration gradient. Note that water moves from the area of lower solute concentration to the area of higher solute concentration, which is how some people choose to think about it. Now let's zoom out and take a look at our U-shaped tube after osmosis has occurred. The free water molecules have diffused to the right through the selectively permeable membrane until the concentrations of free water and sugar are about equal on both sides of the tube. Notice how now that osmosis has occurred, the number of green dots on both sides of the membrane has not changed since the start, but the volume of water on the two sides has been adjusted by osmosis. The solution volume is now much higher on the right than on the left due to the movement of so much water. Now each side has about the same concentration of solute. At this point, when there is no concentration gradient, which way will the free water molecules move? Once the solute concentrations on both sides of the membrane are about equal, the free water will move equally in both directions. We've used this artificial system to explain osmosis. the diffusion of free water molecules across a selectively permeable membrane. Keep in mind that living cells also have selectively permeable membranes, and osmosis is crucial to the function of all cells.
All right, so as the video very nicely explained, what happens with osmosis is it is a gradient. A lot of times I see students wanting to predict the direction of movement going the opposite way because we're thinking more about simple diffusion. But what is establishing the gradient are those free water molecules, which are going to be higher on the side where there's nothing to bind them to, where there's no interaction for that hydrogen bonding. So you noticed in the video, the glucose molecules, the sugar was green, but here it's purple. I use different editions of the textbook. So the way I like to describe osmosis is it is the diffusion of the free water molecules along their gradient, yes, but also it is the movement of water towards the side where the solute is in higher concentration, is another helpful way to explain it. Okay, so this brings us to a property called tonicity, and it is the ability of a cell or a system to either gain water or lose water in response to the concentration of solutes. So what does it depend upon? We need to know the amount of solutes outside of the cell relative to the inside. The thing about tonicity is it always describes the solution. If you have to memorize it at first, please do that. It never describes the inside of the cell, although it is related. The term is a tonic solution. So it's the environment. It's not used to describe the solute. There's a completely different term that we use to describe the solute, but we're learning about tonicity, which is a property in the environment, that extracellular environment. Let's take a look. Now, we can apply 3 different prefixes to the word tonic that describe different situations. And we'll look at the example of the red blood cell in these different conditions so that we can begin to understand it. So the prefix iso means the same. So in the isotonic solution, the concentration's the same on both sides of that membrane. So there will be no change in the volume of the cell because there's nothing to drive it. Similarly to what we just saw at the end of that video on osmosis. The hypotonic solution, hypo means less than. The hypotonic solution is a solution that is in less concentration than what's inside the cell. So what happens here is water molecules are going to move to the cell, and the cell actually gets bigger. We'll look at the pictures in just a minute. Oppositely, the hypertonic solution, hyper means more than, this is a solution or environment that is higher in concentration than what's inside the cell. So what happens here is the water molecules are going to move outside of the cell into the solution, effectively trying to dilute the solution, and the cell shrinks. Okay, so let's focus please just on the top part, looking at the red blood cell. So this is the human red blood cell, which has this shape normally in our normal physiological conditions. It is described as a biconcave disc because it does have this depression in the middle. And this is good. It relates to its structure and function. It allows the red blood cell to squeeze through all the membranes it needs to. It also allows it to carry a lot of stuff inside it in terms, what am I talking about? I'm talking about oxygen, to transport oxygen, because that's the job of the red blood cell. So its normal shape is like this. In the isotonic solution, the amount of water going across the membrane into the cell is equal to what is out. So there is a dynamic equilibrium. Whereas in the hypotonic solution, now there is the solution is a weaker concentration than inside the cell. So the water is going to move from the environment to the cell. The cell is going to get bigger and bigger and bigger and eventually pop. And then oppositely, in the hypertonic solution, now the solution is far more concentrated. Notice the shades of blue in the solution. So it means that the darker blue is more highly concentrated. So the water wants to come out of the cell to effectively dilute the solution. So let me show this on the whiteboard real quick with some numbers, just so that you have a little something to go with. So let me just grab the whiteboard, and Meatball's gonna run away, but that's okay. Okay, sorry, buddy. Just don't jump. It's okay, guy. I hope he doesn't jump down, because then he's gonna knock the board over. Okay, I'm gonna bring this closer to you as soon as I get it all drawn out, but we've got our hypo, the iso, and now the third condition is the hyper. Okay, so we have a beaker that's got a solution in it and a cell. Doing good so far. He doesn't seem to be scared. Okay, so in the isotonic solution, we're just going to make up some arbitrary numbers that fit what's being described. So we have 10 units. The concentration is 10 inside the cell, and it's 10 outside the cell. So that means the movement of water in is equal to the movement out, and there's no change in the volume of the cell. So there's no change in the cell volume. But in the hypotonic solution, now it's less outside the cell as it is inside the cell. So we've got an arbitrary 10 units of concentration in the solution versus 100 in the cell. So the way that the water is going to move is into the cell. And the cell's going to get bigger and bigger and bigger, like a balloon taking on more. Like, imagine you're filling up a water balloon. It gets bigger and bigger and bigger, and then eventually it could pop if providing it doesn't fall off the hose. So here the cell gets bigger. So the cell gains volume, and it eventually will lyse. It eventually undergoes lysis. It pops. The hypertonic is the opposite. So now we have our 100 units in the environment. Remember, it always describes the environment, and we've only got 10 inside. So the hypertonic means that's how I knew to put 100 in the environment instead of in the cell. It always describes the solution. What's going to happen here is the water is going to move this way, out of the cell to the environment, trying to dilute it. So the cell, it's going to lose volume, and eventually it's going to shrivel up. And the membranes will just end up falling apart because they end up collapsed on each other. So here is up close. Sorry for the bright light shining. Oh my gosh, I can't get away. So that is an example using some math, using some relative concentrations to show you about tonicity. Again, always a property of the environment And it, as maybe you now see, it is tied to the concentration of the cell. We need to know that that information is helpful, but it's a property of the solution always. Now, in the plant cell, it's a little bit different, and we're not going to spend much time here because you're in a pre-health professionals biology. It's different because the cell wall of a plant, and the cell wall has strength. It makes the cells more uniform in shape and size. So the way that, excuse me, we have slightly different appearances. The normal condition for the plant is actually in the hypotonic solution. So that's when the plant has its normal looking appearance and is functioning in its normal physiology. It's wilted in the flaccid position. And then the same is true in the hypertonic solution between plants and animals. But it has to do with the cell wall and the pressure between everything inside the cell on the cell wall. And that changes with the vacuoles. Remember, the plant cell has that extra organelle that we don't have, the central water vacuoles. And those are the bladders that hold water for the plant cell. But it provides, when they're full, it provides a good amount of pressure back on the plant, which on the plant cell wall, which allows it to be nice and strong and look normal. So this brings us to the concept of osmoregulation. So now we're moving to tie in the concentration inside the cell with that environment. So it simply is just the control of water balance. And we have a number of control mechanisms and loops in our body that can deal with problems if we have a change in our osmoregulation. I mean, for example, we've all been thirsty, right? That's a biological response that is associated with having too little salt in our bloodstream. Our body figures out there's not enough salt, figures out there's too much salt. I said that backwards, and then makes us thirsty. It gives us the craving of thirst so that we go and drink water so that we hopefully get more water volume back in our body. Now, to deal with the hypo or hypertonic environment, right, our cells have evolved to be able to deal with the movement of water. Okay, so getting back to diffusion before we move on to active transport, those small fat molecules, which is hydrophobic, that means water, fat loving, they can diffuse through. But now we're going to look at the specialized case of facilitated diffusion in a little bit more detail. So facilitated diffusion, again, still is diffusion, but it is needed if something is polar. Only the small non-polar fats are capable of diffusing by themselves. So by default, anything that's not in that category, so anything that's polar or has a charge, it can't go by diffusion. It needs a protein to facilitate that process. And that's exactly what facilitated diffusion is. So it uses a specific transport protein. Still is passive, it still is diffusion. It does not require energy. And that means it is using that concentration gradient. So we have a transport protein for facilitated diffusion called aquaporin. And as you might be picking up from the name, it's associated with water. It is the water pore. So this is the facilitated diffusion channel that allows water to move rapidly across our membranes in or out of the cell. It allows it to osmoregulate. Some water does move by simple diffusion, but aquaporin allows for that process to happen quicker. And this protein, aquaporin, is specific for water. Okay, so let's look at an example of some of the specificity as well as the gradient. So we're looking at a transport protein here that is specific for the yellow circle. And let's look at our concentration gradients. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 circles on outside the cell and only 2 inside the cell. So our gradient is high out here to low inside. So diffusion will happen until that solute molecule has hit its dynamic equilibrium. The triangle should have no bearing whatsoever since it does not interact with this transport protein. Okay, diffusion. Now we need to switch gears and talk about active transport. So I'm going to come back and finish up the rest of that video that we started at earlier in the class in just a moment. But this is when there isn't a gradient and it requires energy. The cell has to invest some energy in order to move whatever it's moving. And the form of energy is a molecule called ATP. We'll look at it. We'll look at its structure in just a moment. So looking at the example of the active transport protein, so we've got, it spans the membrane and it is specific for this green square in this example. The gradient is such that we have 1, 2, 3, 4 molecules outside and 1, 2, 3, 4 inside, or at least before it started. So there is no gradient. There's nothing to facilitate diffusion. So that means this transport reaction needs energy from this molecule. And every time it moves a green square across, it needs more energy. So the way that this channel works, it's like a clamshell. It's open to one side and then the molecule goes in and then it changes shape and opens to the other. Of course, it needs energy to do that. So this is active transport.
The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient.
Okay, so that's the rest of that video that we began with earlier in the module. The first part focused on diffusion and the second half on active transport. So please take a look at this. It has a lot of good information about these processes packed into them. All right, so let's talk about some of the processes to move these molecules that does invest energy or cost energy. So we have exocytosis versus endocytosis. Cyto means cell, exo is exit, endo is within. So exocytosis moves something outside a cell, endocytosis moves something in a cell. So we use exocytosis to exit or export big molecules, like a protein or a polymer, a polysaccharide. Where endocytosis is the other direction, bringing in that large molecule. So for this to happen, both processes do require energy and they use a transport vesicle. So remember back to the endomembrane system when we were learning about the cell, we talked a lot about transport vesicles in that module. We'll review it here. So again, energy is required. It means active transport means that there is an energy requirement. Okay, so we do have two types of endocytosis. There's what's called phagocytosis, and this is essentially when one cell eats something. So that's what phago means, to eat a cell. So the cell does this by wrapping itself around whatever it's eating, and it makes a temporary vacuole. And then we have another kind called receptor-mediated endocytosis that uses a receptor that is interacting with a matched partner. So receptor-mediated endocytosis is a specific process because it's bringing in what binds to that protein receptor. So we'll look at how that happens. So this is phagocytosis. So we've got our cell, and then this is the cell membrane, you can see. And it forms what are called pseudopodium. That means a fake appendage, fake arm, forms two of them, and it's wrapping around this food molecule. And then finally, it will pop off, forming a food vacuole. And then the food is going to be digested with digestive enzymes in the next step. So phagocytosis is a process to move in a lot of material. Vacuoles are bigger than vesicles for the most part, so it's a bigger process. What often happens that we don't see represented in this figure is some other stuff gets in there. This is not a specific process. There's no receptor here. There's no protein interaction. So oftentimes it involves getting other stuff in there too. Okay, receptor-mediated endocytosis is shown here. So here's our receptor, the Y shape, and it binds this purple triangle. I've actually emailed the publisher trying to get them to change the colors and also to eliminate this green solute here. So what happens is the matched protein binds to the receptor, and that sends a signal to the cell. Notice that the part of the cell that is pinching off, it has this other coat protein here. That coat protein is just the cell signal to pinch off there. So notice that when the vesicle is formed, it's surrounded with the coat protein because this is just the inside message to the cell saying, hey, pinch off here, create the vesicle here. So one of the things that bothers me about this figure, and it would be fine if it were correctly represented in phagocytosis, is it brings in this non-specific material. See, we've got this other molecule here that does not interact with the receptor. And yes, that does happen. It does. That's not my dispute. What bothers me is that they don't show that it happens here, because it happens here more because there's not a protein receptor to select what's coming in. So I feel that can be a little bit misbalanced in when a student just looks at those two figures. I want you to understand that with receptor-mediated endocytosis, that this is a specific process because of that protein-protein interaction. Okay, let's play this video on this part of it.
Exocytosis is the movement of intracellular vesicles to the plasma membrane, where they fuse with the membrane and release their contents into the surrounding fluid. This process occurs predominantly in secretory cells, such as mucus-producing cells or pancreatic cells, that secrete enzymes into the digestive tract.
So one important point brought up in this video is that she said that this occurs most predominantly in secretory cells. And that's absolutely true. So that is a cell that releases a secretion. So for example, where would that cell be found? Anywhere in our body that comes into contact with the environment. We have secretions in our oral cavity. We have secretions from our eyes. Right? We have secretions. Proteins are costly for a cell to make. They take a lot of time, they take a lot of materials, and they take a lot of energy. So most cells keep their proteins for themselves. They're not transported outside the cell unless we're talking about a secretory cell, which their job is to do that. All right, this is phagocytosis. Why aren't we working?
In endocytosis, membranes invaginate or pinch in to form a vesicle, moving the enclosed materials inside the cell. This process can take different forms, each involving its own specific cell machinery. In phagocytosis, or cell eating, the cell engulfs debris, bacteria, or other sizable objects. Phagocytosis occurs in specialized cells called phagocytes, which include macrophages, neutrophils, and other white blood cells. Invagination produces a vacuole, which usually fuses with one or more lysosomes containing hydrolytic enzymes. Materials in the vacuole are broken down by these enzymes and degraded.
All right, class, that video showed you absolutely nothing because it was mostly all talking. It didn't show you much of the process of phagocytosis, but that's bringing the material in. Just showed it at the beginning. Let's see if I can get this one to play now.
Unlike phagocytosis and pinocytosis, receptor-mediated endocytosis is very specific. It is triggered when membrane receptors bind to specific external molecules, such as protein cholesterol complexes or proteins bound to iron. Membrane vesicles pinch off, and the external protein and its cargo are brought into the cell.
Receptor-mediated endocytosis. All right, we're going to move to the next part on energy, which will wrap up with enzymes. So energy is just the ability to do work. And we're going to talk a little bit about what work is, but we have two types of energy. There is kinetic energy, which is always associated with movement. Kinetic energy is the energy of movement, whereas potential energy is chemical energy. It's based upon the potential to do work. And that's ties to it gets down to the chemical bonds and our big molecules that have a lot of those chemical bonds in it. So photosynthesis is a great example of kinetic energy, and it utilizes the sunlight to move electrons. Whereas examples of potential energy are in those big macromolecules that we have, specifically in the chemical bonds. Now, you've probably heard of the term thermal energy, and this is the energy associated with random movement. So it is a type of kinetic energy since we're talking about movement. It's different from heat. Heat is the transfer of that thermal energy from one object to another, where thermal energy is the actual energy associated with the movement. So chemical energy and potential energy, those are the same things. You can use those words interchangeably. So when a chemical bond is rearranged, there is a potential to generate energy. It doesn't mean it's going to happen every time, but there is potential. And we'll learn more about some of these different types of chemical reactions that do this. So it is potential energy that is the nuts and bolts of a living organism. It's the most important type of energy because it's what is used to power all of the cellular activities. Now, thermodynamics is a discipline of science that simply studies energy as well as how energy changes. We use the word system to describe what we're studying. So the cell is a system, where surroundings is the environment. So the extracellular environment would be an example of the surroundings, or we could look at the level of the organism as the system. The extracellular environment would be, or the outside environment would be everything else. Now, we do have two laws of thermodynamics, and they're actually pretty simple. The first law of thermodynamics, which is the conservation of energy, It simply states that energy is constant in the universe, like the quantity of it is constant in the universe. It may change forms, but it doesn't just go away or appear magically. So it changes forms. The second law of thermodynamics is tied to a property called entropy, which is just disorder. And what the second law states is that there are going to be energy conversions that happen to increase disorder. So think about, what's a good example? Okay, so think about if you take a pot of water, to keep our example going, and you put it on the lid, on the stove, but now you have a tight fitting lid on it. And you apply heat and you get the water nice and boiling. What happens if the system is closed? If you have the lid on the pot, that water is going to be boiling like crazy and perhaps splashing. It could even blow the lid off. But if the system is open and didn't have a lid on it, then the water would just evaporate away. So that's kind of a different example of how to apply the disorder in the universe. So there's more disorder in that closed system because it doesn't provide the opportunity for the energy to transfer somewhere else or in a different form, right? It can't transfer from, the energy can't really transfer when the water goes into that vapor phase if it's all closed off. So entropy is that measurement of disorder or chaos in the universe, but it really needs to be focused at the system too. Okay, so let's look at what might seem like a really weird comparison. We're going to look at the thermodynamics of the combustion car. So, you know, your old timey car, not talking about a battery car or e-car. And we're going to compare that with cellular respiration, which is the metabolism at the level of the cell. There's so many similarities between these, so it's very relevant to compare them. But surprisingly, the cell is way more efficient at this process than is the combustible engine of an automobile. Now look at both of their starting materials. They both use the same material of gasoline and molecular oxygen. How do I know it's molecular oxygen? Because there's two. That means molecular oxygen. The waste products are even the same, carbon dioxide and water. They both release heat. So that is unused energy from this reaction that is being released to the system. Let's look at it. So gasoline and oxygen are converted through the engine. So when my car's sitting in my driveway right now, it's got lots of potential energy, but zero kinetic energy because it's not on. But as soon as I turn it on and press gas into the engine, that's going to power the engine to move, which drives the, I don't know, the axle, the rotor. It drives the part that moves the wheels. And now we've got kinetic energy. And then carbon dioxide and water are going to be released as the engine process goes on. Now, in the cell, it's very similar, same starting material. We do have a series of chemical reactions that happen within the cell, which we're going to learn about in our next module. And we have some of the energy released in the form of heat. The thing about us is our body is really smart. We don't just waste all that heat from cellular respiration. That's part of what gives us our internal body temperature. So we benefit from that so-called wasted heat. So the energy conversions are comparable. Just the cell is way better. It's much more efficient than is the engine. So this is cellular respiration, which is the content of our next module, the module after next. We get into quite a bit more of it. This is a really important set of chemical reactions that we'll study because it provides the energy needed to support life. So chemical reactions can be grouped a number of different ways, but we're going to look at grouping them by energy. So the exergonic reaction releases energy where the endergonic requires. Same prefixes, exer to exit or export, ender within or to bring in. So some of our reactions need energy in order for them to happen. Exergonic reactions release energy, and then the cell can save that energy. It banks it up and saves it, and then it can be used for something else. So it happens when the covalent bonds of those macromolecules are broken down. So this ties in the hydrolysis reaction we learned about earlier in the semester, as well as the reverse reaction of dehydration synthesis. So when we burn wood, there's energy released from the glucose that's in that wood, and we see it and feel it in the form of heat and light. Cellular respiration is an exergonic reaction. It's a great example of an exergonic reaction. It's the point of it. It's to generate energy. So after all of the chemical reactions of cellular respiration occur, there is leftover energy that is banked and saved by the cell that can be used for other purposes. It does have many different steps in it that we'll learn about. And it's a process that releases energy slowly. And it does this in the form of producing or releasing energy from this molecule called ATP. So let's look at a graph of the exergonic reaction, a simple one. On the x-axis is time, or progress of the chemical reaction would be appropriate as well. On the y-axis is energy, potential energy, and it increases as we move away on the axes for both of them. We are looking at the reactants of a chemical exergonic reaction versus the products and plotting how much energy is associated with either the reactants or the products. The chemical reaction is represented by the arrow. That's what arrows mean. It means chemical reaction. The key about the exergonic reaction, there is energy to exit, is the reactants have more than the products. So we have a defined amount of energy associated with the reactants, which is more than is associated with the products. That means the difference is released. It is exiting this chemical reaction and can be saved and used for a different one. So in the exergonic reactions, the products are low energy, where the reactants are high energy. And the difference is what is exited. Oppositely, the endergonic reactions, they need energy to start. So they produce products that have energy. But they start with molecules that have very little energy, which is why they need energy from somewhere else. So let's look at that graph. Photosynthesis is a great example of the endergonic reaction, and that input of energy comes from Mother Nature. It comes from the sun. Okay, so the endergonic reaction is different from what we saw from the exergonic. Now the reactants are low energy and the products are high energy. And the difference is the amount of energy that needs to be put into this reaction in order to make the products. We can't just make high energy products without having the right amount of energy to create them. So endergonic reactions need energy, that difference in reaction energy between reactants and products, in order for the formation of the products to occur. So oftentimes endergonic and exergonic reactions are coupled because exergonic reactions have energy to exit, which then can enter the endergonic. So that's what we see, is that there's thousands of endergonic and exergonic reactions that define metabolism. And that simply is the sum of all those chemical reactions associated with breaking down materials that we eat and then making the different materials that, that we need, and all the work that the cells perform. So a metabolic pathway is really just a series of chemical reactions to make a molecule, or again, to break it down. So we're talking about monomers being built together to make polymers, or polymers being broken down into their monomers. So energy coupling is what we see with these endergonic and exergonic reactions, where we take the energy exiting the exergonic reaction to power the endergonic. And the energy is stored in the form of ATP. So one of the things I've seen described is sometimes we don't recognize that there is a way to store this energy. It doesn't mean that the reactions are happening right together at the exact same time and like a relay. The cell can bank the energy and save it up and use it for what is needed. So here's this molecule, adenosine triphosphate. Tri means 3. So it's got 3 phosphate groups linked to adenosine. And it's used for pretty much every form of cellular work by all living organisms. So it's got a triphosphate tail, and the energy movement happens when a phosphate group moves. We'll see the cycle. So now we're looking at the hydrolysis of ATP, which means breakdown, right? Go back to our earlier chapters where we learned about hydrolysis and dehydration synthesis. It's the exact same reactions, and we really need to understand them still now. So ATP is hydrolyzed, which releases a phosphate group. Now one of these phosphate groups has moved away, and we now have adenosine diphosphate plus a free organic phosphate group that then can be added to something else through phosphorylation. That's what phosphorylation means, is to add a phosphate group. Okay, so we have 3 main types of cellular work. We've got chemical, we've got mechanical, and then there's transport work. We've spent some time already in this module talking about transport work. And ATP is used again for every single one of these. All right, so we're looking at pretty simple examples of the 3 forms of cellular work. So an example of what we just looked at with the hydrolysis of ATP is a good example of chemical work, any type of chemical reaction that is involved in energy. So we have our reactants and our products. So looking now at the example of transport work, we have a transport protein that is using energy from this phosphate group that came from the hydrolysis of ATP to power the movement of the solid molecule against its gradient. And then in the example for mechanical work, we're looking at what happens with muscle, leading to a contraction of a muscle. So this is a muscle fiber up here, the pink. And then we have a different type of muscle fiber down here with these blue circles, but they are connected through a chemical bond that is triggered to move through the hydrolysis when this phosphate group moves off. So once this phosphate group moves off, it causes a contraction and movement of the muscle. So this is the complete ATP cycle, beginning with adenosine triphosphate, the hydrolysis of ATP. So adding water across that bond releases a phosphate group, which then can be used, that energy can be used for anything the cell needs. If we complete the cycle and add on a phosphate group to adenine diphosphate, so that would be phosphorylation. Now we have completed the cycle through the formation of ATP. But to create ATP, we need energy. to break ATP, we release energy. Okay, so the last section is going to focus on enzymes. And enzymes are just so critical for metabolism. In chemistry, we teach that catalysts speed up the rate of a chemical reaction. And that is absolutely true. An enzyme is a catalyst made by the body. It's incorrect to that term is functionally different for us when we think about it from a biological perspective. It is true that enzymes speed up the rate of the reaction, but the truth is that many of the reactions of metabolism simply don't occur fast enough to support life without the enzyme. So it's not just a matter of making it happen faster. It's a matter of creating the conditions that support life. And enzymes are absolutely essential for that. They do it by reducing what is called the activation energy. And this is an energy barrier. So, say you're doing, say you're going downhill skiing, but you first have to walk up the hill to get there. That getting up, that's your activation energy to ski the lift and do the fun part. Okay. So enzymes are those biological catalysts. They are mostly made up of protein. There's a few examples of RNA-based enzymes, but by far they're protein molecules. Now, they are not used up in the reaction. They facilitate the reaction, but they do not become part of the product. What that means is that the enzyme after the reaction is free to be used again because it's not consumed by the reaction. So they, again, work by lowering the energy of activation. So we need to look at a picture here. All right, we got quite a few things on here. We're going to begin at the bottom left of the figure. So we have the energy of the reactants and the energy of the products. Right now, please don't think about the exothermic or exergonic or endergonic reaction. We're just going to focus on the activation energy. So the gray column here represents that energy barrier. So we've got time or progress of the reaction on the x-axis and then increasing energy on the right. So in order for the reactants to be converted to the products, which is a hydrolysis, we're starting with a dimer and breaking it into two monomers. The chemical bond is broken, but we have to get over this barrier to get there. So what the enzyme does is it reduces this barrier, and it's doing it by simply holding the reactant. This is a type of enzyme that literally holds the reactant in place, and that makes it much easier to hydrolyze that chemical bond, which would free up the products simpler. So that's the difference. That's how they work, I should say. This is a type of enzyme called let me type it up here for you, a substrate enzyme. There's another category of enzymes we'll talk about when we get to cellular respiration, but this is properly called a substrate enzyme because it's holding the material so that the chemical reaction is facilitated. Okay, let's take a look at this video.
If exergonic reactions occur spontaneously, what keeps molecules from breaking apart and cell chemistry from racing out of control? For any reaction to occur, even a downhill reaction, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, E sub A. This required energy input represents a barrier that prevents even energy-releasing exergonic reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules referred to as the enzyme substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Okay, let's back up for a moment so I can go over something else. So looking at this top part of the figure now, which is a summary, it's showing us a line graph for both conditions, with and without an enzyme. Notice that the energy of the reactants with and without the enzyme is the same. The energy of the products with and without the enzyme is the same. All that changes is this energy barrier. The black is without the enzyme, the red is with it. So it reduces that activation energy, which makes it favors the formation of the products. So when we're talking about an enzyme, a substrate enzyme, we don't call the starting material a reactant anymore. It's properly called a substrate. So when you see that term, substrate, even if you're not told, you should associate that with a chemical reaction that requires the enzyme. So enzymes are selective because they're proteins. And oftentimes we see, really great selectivity. So because we're talking about protein-protein interactions, this leads to the specificity we would expect. So it is that three-dimensional shape of the enzyme, of the protein, that determines its function, its specificity, just as we've been talking about all semester long since we began talking about proteins. It's always about the three-dimensional interaction. So the reactant is called a substrate when we're using the enzyme. And the place on an enzyme where the substrate fits, that is called the active site of the enzyme. So the active site is part of the enzyme where the substrate binds. So the specificity between an enzyme and its substrate is dictated by what fits into that active site. If it only lets one thing in, very, very specific. But if it allows more than one thing to fit, which sometimes that happens, then it's not quite as specific. Okay, so let's run through the cycle. So we're looking at our enzyme, and this part here is the active site. And this is an enzyme called sucrase. The ending ASE tells us it's an enzyme that acts on sucrose. Many enzymes do have the ending ASE. Not all, but many. So if you see it, please think enzyme. The substrate is the sugar sucrose, which is composed of two different monomers. So sucrose binds into the active site of sucrase in a very specific way, and that allows for the hydrolysis of the covalent bond that holds together the two monomers. So we need water for hydrolysis to happen, and that water splits apart. Hydrogen goes on one of the sugars, the OH goes on the other, so everybody's happy with their octet rule. And now the products are released, which would be glucose and fructose. And the enzyme, providing it has not been damaged, is now free to be used again. The enzyme does not become part of either product. It is not used or consumed by the reaction. It simply facilitates the reaction by holding the substrate in place. That lowers the activation energy to make the products of glucose and fructose. Now, our enzymes do function under optimal conditions, and every enzyme has its own conditions. But when we're talking about the human body, if we're talking about our enzymes, it's actually quite simple. Well, they function at our physiological conditions. That's how it works. We do know that temperature is associated with molecular motion, but temperature, heat is never an enzyme. And the reason why is because it doesn't meet the definition of an enzyme. If you take the pot of boiling water off of the heat source, it's going to cool down. Enzymes are not used or consumed by the reaction. They are not involved in the formation of the products. So temperature will make reactions occur faster because it increases molecular motion. That increases the chances of a chemical interaction, but it is not an enzyme. So there is an optimal temperature for each enzyme, and for us, it's our body temperature. So they're usually optimized to function between 35 to 40 degrees Celsius, which 37 degrees Celsius is 98.6. And the same is true with pH and salt content. Enzymes function best when they're like our blood, when they have the same pH or the same salt concentration of our blood. Now, cofactors are required for many enzymatic reactions, especially in metabolism. So all a cofactor is it requires something to go with it, to cohabitate, if you will. So these are non-protein helpers. And what they do is they do bind to the active site. So that's why they're called a cofactor, because they are required to get that correct shape of the active site. So some cofactors are made of different metals. So they're inorganic, things like copper and zinc and iron. Whereas we have others that are made out of vitamins. So those are called coenzymes because they're made from an organic material. And specifically, many coenzymes are derivatives of vitamin Bs. This is the reason behind the like those health spas where you can go and get a vitamin B infusion. The thought is to provide more vitamin B, since some of the coenzymes associated with metabolism are made from vitamin B. So they're vitamins. Okay, so enzymes can be inhibited with what's called the inhibitor. And actually many different drugs are inhibitors of different processes. We have two types of inhibitors. There's the competitive and then the non-competitive. And it's a very fundamental difference, a simple difference. The competitive inhibitor is going to the active site. It is competing for the active site. Where the non-competitive inhibitor, it does not bind the active site, but it changes the shape of it. So it binds somewhere else than the active site, but does change the shape. It inhibits. So the outcome is the same, inhibition. Let's look at some pictures. Okay, so here we've got the enzyme. The substrate is blue, the enzyme's purple. You can see the shape of the active site. And then we have a binding site down here. A competitive inhibitor is getting into that active site. It prevents the substrate from reaching or interacting properly with the active site of the enzyme. The non-competitive inhibitor binds at a different location on the enzyme outside of the active site, but it changes the shape of the active site. So even when the substrate is available, once the non-competitive inhibitor is interacting with the enzyme, it can't work. The shape does not match. So the chemical reaction is inhibited. So we see there are actually many enzymes inhibitors that our body uses to regulate and control metabolism. And this all occurs through what is called regulation or feedback. Feedback inhibition would be what we would call it with an enzyme inhibitor. So let's look at an example. And we're almost done with this lecture. Okay, so we've got a sequential enzymatic reaction to begin with. The product is D. We begin with A. We have enzyme 1, we have enzyme 2, and we have enzyme 3. There is a described feedback inhibition loop between the product and enzyme 1. So let's run through this. So A binds into the active site of the first enzyme, and it produces product B. B binds to the active site of the second enzyme and produces C. B and C are both intermediate molecules. So they're not this product, but I'm willing to bet you that they're used for something else that the body needs. Our body doesn't just produce things and then not use it for something. So B and C are going to be used for something else. C interacts with the third enzyme, which has its matched active site. Note the differences in shape of each of these active sites on the enzymes. And now we finally get our product, D. And when we have enough D, it sends a message all the way back to enzyme 1 saying, hey, we have enough, we don't need you anymore. We can turn off this pathway so we don't waste materials. All right, so examples of enzyme inhibitors that are used in pharmaceutical would be ibuprofen's a great example. It inhibits a molecule called, or group of molecules called the prostaglandins, and these are associated with inflammation. So ibuprofen's actually great to take for anything with inflammation. So like if you sprain your arm and it's swollen, ibuprofen is a much better choice to take, providing there's no drug allergy, than say Tylenol, acetaminophen, because ibuprofen actually interferes with the process that causes the pathology. It reduces inflammation. So it's an anti-inflammatory as well as treating pain. Acetaminophen just treats pain. There's different classes of blood pressure medications that are enzyme inhibitors. For example, the ACE category of blood pressure medicine. There's antidepressants, many antibiotics. There are even antivirals that are inhibitors. So one category are the protease inhibitors that are used for the treatment of HIV. And then we've got examples of pesticides and even poisons that are enzyme inhibitors. So it's widely used throughout many, in nature, naturally, as well as in many industries. All right, so the slides back here are those extra concept slides I'd like for you to really know inside and out. So it's not a matter of just memorizing the terms. Please be able to apply this information to a scenario or extract information out of a scenario. So we've got some of the bigger concepts that we've talked about. Okay. Now what I'm going to do before I pop off of here is I wanted to go back to that complete video on diffusion and active transport, and I'm going to play that whole thing. I do think I've gotten most of it in the different segments throughout the lecture, but just in case, I would like to play the whole thing again so that you have it for those of you that don't have the textbook. Okay, share that screen again.
We get all our energy and organic molecules from food. Before we can use the molecules we eat, They have to enter our cells, starting with the cells lining the small intestine. Let's zoom in to the surface of a cell. The plasma membrane is selectively permeable. Some molecules can move across it, while others cannot. How do materials enter and leave cells? Lipids, such as these yellow molecules, can dissolve in the lipid bilayer. Notice how they move down their concentration gradient, from where they are more concentrated to where they are less concentrated. This is an example of diffusion. Diffusion is a form of passive transport. It does not require energy from the cell. Most molecules can't cross the lipid bilayer. Here, the sugar fructose moves into intestinal cells by facilitated diffusion, moving down its concentration gradient through a transport protein. Facilitated diffusion doesn't require energy from the cell, so it's also a form of passive transport. Water crosses the plasma membrane by facilitated diffusion, or by diffusing across the lipid bilayer directly. The diffusion of water across a membrane is called osmosis. The sodium-potassium pump moves ions against their concentration gradient, from where they are less concentrated to where they are more concentrated. This requires energy from the cell and is known as active transport. Energy from ATP is used to move sodium ions out of the cell and potassium ions in. Another type of active transport is cotransport. Here, both sodium ions and glucose move into the cell through a cotransporter protein. Sodium ions move down the concentration gradient created by the sodium-potassium pump, and glucose moves against its concentration gradient. Now let's move to the other side of our intestinal cell. Materials can be exported in vesicles that fuse with the plasma membrane and release their contents outside the cell. This process is called exocytosis. In endocytosis, the plasma membrane pinches in, forming a vesicle that contains material from outside the cell. On this side of the cell, we can also see oxygen and carbon dioxide diffusing across the lipid bilayer. Cells use all these processes to get what we need.
Okay, well, that's a wrap on the Module 8 lecture. I hope you enjoy the rest of your day or night, wherever or whenever you are viewing this. As always, please feel free to reach out by email. Take care, everybody. Bye-bye.